Hi, everybody. I'm Arsalan Arif for Endpoints Webinars, and our topic today is Dell like never before, fourth generation of Dell kits, sponsored by Wuxi AppTech. Before we begin, I'd like to introduce our panel of experts who will be joining us today. First up, Alex Satz, the Senior Director of Dell Strategy and Operations at Wuxi AppTech. Next up, Nushka Shammer, the Head of Dell Lab Operations Europe at Wuxi AppTech. And finally, Ken Westover, Associate Professor of Biochemistry and Radiation Oncology and the Director of Clinical Innovation at UT Southwestern. Now, if you have any questions during today's webinar, please hit the Q&A button at the bottom of your screen as soon as you can think of the question. This webinar will be available on demand tomorrow to rewatch or to share it with your colleagues. And from here, I'm going to pass it off to Alex to kick things off. Alex Satz.
Okay, let me share my screen and put it into the proper mode. And I'll just ask if everyone can hear me, if I'm coming through.
Loud and clear.
Perfect. Okay, yeah, so thank you very much for the introduction. And of course, I'd like to thank all of you who have, so to speak, signed in or called in order to listen to us talk about our fourth generation of Dell kits. We have about 40 to 50 minutes worth of presentation, then followed by a Q&A. So I thought I would first go through what the different topics would end up being. So first off, we're going to discuss why we think the self-service kits are different from the way that Dell screens have been done in the past. Then we will discuss work that we've done to give you the best chance of finding hits that are either drug-like or lead-like. Then we will discuss what we call the frequent hitter index. So we, of course, at Wuxi, we're experts at looking at the Dell screening data and finding the best hits, but we understand that many of you who would run a screen would be doing it for the first time. So we've set up a very fast and easy method for you to be able to weed out the bad stuff and be able to focus on the good stuff. Then, of course, when you run a Dell screen, you have a lot of data that comes out of this screen. And can we use a deep learning approach in order to get the most value from your screening data? So we'll discuss that, and we'll talk about a little POC that we've done. Then my colleague Nushka will talk about the 5-minute QC. So when you do a screen in your own lab, how do you know the screen was done correctly before you send the samples back to us? Well, we will show you the methods that we have come up with. And then, like you who are watching this seminar now, people like Ken watched this perhaps a year or two years back. And they attempted this, they got a Dell Open kit and they tried it. And he's going to tell you about how that went. And so, yeah, I think you'll be very curious about that story. Okay, so why do we think that the self-service kits are different from, let's call it the more traditional way that Dell has been done in the past? So there is such a thing as full service Dell. We call that Dell Pro for short. And in this case, you send a target to us and we screen it. We look at the results for you and we help you pick the hits that you want to follow up. Now, this method works great, but it does have a few flaws which the self-service approach addresses depending upon what is important to you. So for the self-service kits, we send the kit to you and you physically do the screen in your own lab. So first, if your protein is very sensitive or there are proprietary ways in which you like to handle the protein, which you don't want to share with us at Wuxi, then the self-service kit ends up being perfect for you because you can do the screen in your own lab. And you don't need to tell us how you did the screen. So if you develop new ways or methods to do a Dell screen, you can keep those a secret in your lab. Also, because the screen is done in your lab, you don't need to tell us what the target is. And so for many project teams, this is an important point because they don't want to share their targets with anyone outside of their group. Lastly, when a screen is done physically in your lab, that does lower the cost as well. And this is particularly important for academic labs who would like to use the Dell Open kits, which are essentially free. So because the screen can be done in their own lab, that means that we don't have to charge them. We can just send them the Dell Open Kit, and they can do a Dell Open Kit, and they can find hits at a very low price, essentially free. Okay, so the next question, of course, is how does this process work? How do we get the Dell Open Kit or the Delight Kit? And how do we do these self-service screens? So let's start with the workflow shown here on the left. The parts that we do at Wooshi are shown with the blue dots, and the parts that you do in your own lab are shown with the yellow dots. So the first thing is we make the kit. So we produce the Dell Open Kit. You can go online and you can order this kit, and like I said, it ships to you at no cost, except for perhaps the shipping costs. Then in your lab, you have to physically do the actual screen. We have protocols for this, videos for it. There are many people who have run the screens in their own lab, so it is certainly something that you can probably do. When this physical screen is done, the wet lab samples are then sent back to us, and we are going to do the PCR and the sequencing, and then we generate all of the data from your screen, and we share all of that data with you, except we don't share the chemical structures. So you will get the physical profiles of your hits. You will know a lot about these hits, but you won't know what their exact structures are. You then need to look at this data set and decide your top 5 hits, the hits that you would like us to synthesize for you. We then resynthesize these hits for you, and we send the hits to you, but you still don't know what their structures are, but you are now free to test these hits in your own lab. So whatever assays you have in order to see if these hits provide value to you, you can use. And if you think these hits are good, you just tell us this, and we provide all the structures to you, and we provide those structures to you at no cost. You are then free to publish those structures, patent them, sell them, discover a drug. There's no reach-through claims or milestones from Wooshi. There is a bit of a catch, though. This is called Dell Open, and the purpose of Dell Open is to share data. And what that means is that you need to tell us, when we share the structures with you, you need to tell us what the target was and give us permission to publish your screening data without the structures shown on the Dell Open website. And the purpose of that is so that different academic groups can then go and can look at this data and possibly work with you on it. So it's all about being open, publishing more, and sharing data. Okay, so the purpose of a Delight Kit is a little bit different from Dell Open. Dell Open is for academic labs. Delight is for discovering drugs. So first you have to purchase the kit from us, and then we send the kit to you, and you again physically do the screen in your own lab. You then send the samples to us, we do the sequencing, and again, we then share all of the metadata with you from this screen. And the purpose here is that we share all the data of the screen with you without the structures, but that should be enough data for you to decide if the screen worked well. And if you decide that the screen worked well, then you pay an additional fee and we unblind all of the structures. So what that means is that we might send you a list hundreds of thousands of structures long, or even longer. So this is a very long list of all the potential hits against your target. You then choose which hits you want to follow up. Anything you find is owned by you. Again, there's no milestones, there's no reach-through claims, nothing along those lines. Okay. So has this been successful thus far? Have many people ordered these kits? And the answer is yes. We've been having these self-service kits for about 3 years now. And within that time, we have delivered over 680 kits. Now, like I said, we share the metadata of the screen, but then there's a fee that you have to pay in the case of Delight to unlock all of the data and to actually see the structures. We've unlocked almost 50 million structures. So we take this as a really good sign that the results of these screens have turned out quite well. Now, you might have a concern if we've unlocked 50 million structures is there anything good left, right? So in the last 3 years, have other groups used these kits, gotten all of the good stuff, and now there would be nothing left there. And the goal of this next slide is to tell you that no, that is not the case. There's a lot of structures contained within these self-service kits, and those 50 million structures only account for 0.003% of the total number of structures in the kit. Also, and this is an important point, in the world of DEL, a single chemist can make millions of new structures in the span of a month. And so every month we make more DELs, and every year we make a different generation of these kits. We're adding more and more DELs to these kits, or at least different DELs. So as I'll talk about very soon, the 4th generation kit is full of new DELs with brand new structures that no one's ever seen, no one's ever screened. And so again, there's no worry about people finding all of the good stuff from previous screens. So I did say that we're constantly making new DELs, and what's our goal when we make these DELs? And one of our major goals is to increase the chances that when you do find a hit, that it would fit into that lead-like space or into drug-like space. So let's talk about that for the next few slides. So first, when you look at the 4th generation DEL self-service kits, the Delight kit has 58 DELs in it, and it has about 11 billion different structures. And DEL Open has 27 DELs in it, and it has about 4.2 billion different structures in it. So both these kits have a lot of potential structures in them. So let's more directly compare the 3rd generation Delight kit with the 4th generation Delight kit. So this is the new kit. So what we notice if we look at these two charts is that in the 3rd generation kit, we have a lot of DELs which have 4 cycles of chemistry and only a few that have 2. In the 4th generation DEL kit, we have many more DELs that contain only 2 cycles of chemistry and fewer that contain 4. The important point here is that structures that come out of a 2-cycle DEL tend to be more drug-like and more lead-like, whereas structures that come out of a 4-cycle DEL, these hits tend to be physically larger and tend to be less drug-like. So one way to sort of understand what that means is what I'm showing here on this plot. This is a 3-dimensional representation of the chemical space contained both by the 3rd generation kit and the 4th generation kit. Now, the 3rd generation kit, every structure is shown as a little green dot. And for the 4th generation kit, every structure is shown as a little blue dot. And what we see is that there's a lot of overlap between the 3rd generation kit and the 4th generation kit as shown here. However, we now have this new chemical space being accessed only by the 4th generation kit. And the question is, what are the structures contained within this new space that the 3rd generation kit doesn't cover? And the answer is these are the structures that are coming from these 2-cycle DELs that are fitting into this more lead-like and drug-like space. And that's why they're so separated from the rest of this space as shown here. So I know as a chemist to look at those 3-dimensional chemical space plots can leave you wondering, well, I mean, what are the actual structures? So I'm showing you 2 examples here. One is of a 3-cycle DEL, which is now in the 4th generation kit, and one is of a 2-cycle DEL. So for the 3-cycle DEL, what we see is that 80% of the structures contained within this DEL fit within Weber's rules. And for this 2-cycle DEL, 65% of the structures are contained within the rule of 5. So these are the kinds of new DELs that we have made, which we have put into the new Delight kit that are filling up that new space that previously wasn't covered by the 3rd generation kit. We can also inspect this by looking at the distribution of all the structures contained within the 3rd versus the 4th generation kit. So the distributions for the 3rd generation kit are shown in red, and the distributions for the 4th generation kit are shown in blue. And if you look at the molecular weight or the heavy atom count, you can see that the distributions are shifting to the left. So if I eyeball this roughly, I would say that the heavy atom count is shifting about 5 heavy atoms to the left. And that is, of course, exactly what you would like to see if you want a kit which is going to have a better chance of providing you with a more drug-like or lead-like hit. We see that the fraction of sp3 is shifting to the right, so that's good. We see that these structures are actually more rigid as well. So the number of rotatable bonds is shifting significantly to the left. We see that the log P is shifting a little bit to the left. The PSA has a substantial shift to the left, and hydrogen bond donors are shifting substantially to the left as well. So these are all things that you want to see if your goal is to discover more lead-like or drug-like hits. So we at WuXi, we're experts at looking at the data and determining which hits aren't worth time or effort to follow up, or at least might be of a higher risk. And one of the things that we have access to, of course, is all of the data that we've collected in the past. So we've run many hundreds of different screens. And we know that there are certain structures which end up repeatedly showing up in certain screens. And what we've determined is that such hits that aren't unique for a particular screen probably aren't worth following up or putting in further effort on. So the question is, could we set up an easy way where we could just provide you with a couple of different numbers from an index, and you could, as successfully as we do, make the choices about what hits you would like to follow up? And for that, we've created this frequent hitter index, which runs from a number of 0 to 5. So when you unblind the DELIGHT data, or you look at your Del Open data, every structure shown will have this frequent hitter number assigned to it. And if the number is 0, it means that that particular hit has never shown up in any screen in the past. But if the number is 5, it means that structures like these show up a lot in a lot of different screens, and you might not want to follow up that hit. So we ran a little experiment. We took a self-service screen, and we, as the experts, we looked at this data. We looked at all of the historical data that we have, and we decided which hits we would want to follow up. And we show those hits here as single data points as a circle, and these are shown with the more vibrant colors, so whether it's green or blue. And the hits that we wouldn't want to follow up are sort of grayed out, as you can see here. We also took this data and we plotted it using the index that I talked about on the previous slide, whether these hits were showing up a lot against other targets or whether these hits were showing up a lot in other NTCs, control runs. And what we found is, using just 2 different numbers, if you focus in on the hits that show up in the quadrants 0, 0 or 0, 1, you would be choosing hits that are exactly the same or very much like the hits that we would choose as the experts in the field who have access to the entire data set. So our goal here is so that you, as someone who has never run a Del screen, can receive this data set and can very quickly, just using 2 simple index numbers, be able to assess this data in the same way that we as experts would. So when you run a Del screen, you generate a lot of data. And we've always wondered, do we do the best job you can really do looking at this entire large data set and finding the best hits from it? So in the next few slides, we'll try to address if we could use deep learning in order to look at this data in a more effective manner. So what we start off with is the Dell data itself that comes from the screen that you have run. We can then train upon this data in order to create a predictor, which we can then use to interrogate a database external of compounds that you could just purchase off the shelf. Now, what's nice about this is that when you purchase compounds off of a shelf, you're going to get them faster. They're going to cost a lot less than having to make them from scratch. And you can focus on things that fall within that lead-like space. So you can say, well, I want things that have a C-log P of less than 4, molecular weight less than a certain amount. So I want to focus on really small hits. So that would be the workflow. And the question is, does it actually work? Well, let's run a little proof of concept test. We will take a target, which has been around for quite some time, and we will run a self-service screening kit on it. We will find hits the normal way, and we will also see what kind of hits we can find using the deep learning methods. Okay, so we take our target and we screen it with our self-service kit, and we plot the data out as shown here. So this is the, you could call it the old-fashioned way of doing Dell and looking at a Dell screen. So we have our different Dells shown here on the x-axis, and we have our log of the enrichment values shown here on the y-axis. And what we've done is we've almost randomly chosen 4 different hits from 4 different Dells. We resynthesized each one of these hits and we tested them. And we found indeed that all 4 hits were active. We see that some of these hits, you know, they aren't necessarily large hits. They're relatively small. We do find some other hits that have an interesting structure. They are quite potent, although they do break the rule of 5. They're getting a little bit large in mass. So, you know, all in all, though, the result of this effort seems to have worked out very well. Now let's see what the machine learning does. We classify all of that data. We then interrogate external databases, and we order the different structures at a very low price. So each one of the structures that we order, we can purchase for less than $100. We focus in on compounds with a very low mass, so it's fitting this lead-like space that we would like to focus on. And we find that we end up with about a 5% hit rate. So these are 5% of the hits that we follow up are indeed active. We see that the structures we find are different from the structures that I showed on the other slide. And they were indeed active, although not as potent. However, these compounds do come at a much lower cost, and their molecular weight is lower. Okay, so this slide summarizes the output here. So if we look at the plot on the left in the dark blue lines, we see the masses of the hits that we followed up using the deep learning method. And in the light blue, we can see the C-log P of these hits. So we see that it's under 400 grams per mole and well under a C-log P of 4. So it's fitting into lead-like space. And in dark blue, we see that we were able to acquire these hits in 4 weeks. Of course, if you have to synthesize a hit from scratch in the more old-fashioned way, then your timelines are going to be closer to 7 or 8 weeks. And of course, the cost for these hits is much lower because they're sitting on a shelf. And so the cost for each one of these hits ends up somewhere in that $50 to $200 range. Okay. So when it comes to a Delight kit, we have partnered with an external company who has a lot of expertise in doing this. So if you want to make use of the deep learning with a Dell Open or with Dell Pro, this is work that we would do, but for a Delight kit, we have a partner who has a lot of experience in this field. Okay, So the next topic is the 5-minute QC. My colleague Nuska will run through those slides. So Nuska, just tell me
Thank
when
you.
I should advance.
Thank you, Alex. So yeah, please advance to the next slide. So basically what we actually wanted to provide here is a brief information how you can actually test by yourself when you are performing the experiments, if your samples are, let's say, good enough and it makes sense to proceed to the next step. So basically, when you're done with affinity selection using self-serve kit, usually then you send your samples to us and we perform the very detailed sample quality control where we use qPCR, very careful, highly standardized protocols. And of course, to do that, you also have to be very trained and skilled so that you are always practically redoing in the same manner. Everything has to be very standardized. So still, however, we often got a question from the clients that they would like to check how their samples looks like while they're performing affinity selection in their laboratories. So in our third generation, that means current generation of the Dell Light Kit, we actually included so-called colorimetric card, where based on the band intensity on your DNA gel, actually you can get a rough feeling how many copies are there in your samples. Because practically we spiked in the DNA, which also practically because these DNA particles, let's say, or fragments are running at different sizes, depending on the condition in which you were using, we can also detect cross-contamination. So, I mean, as you hear, practically I already need that quite some time to describe how this works. So that means also that you will need quite some time to execute this in the practically in your lab. So, I mean, it brought a lot of advantages. So many clients decided to use it and to practically find out what are the levels of cross-contamination and how many practically copies they have in their samples. Disadvantages, time-consuming, you need equipment, and results often depend on the gel and staining used. So to avoid all these headaches, we actually invested a lot of time and creativity, and I'm proud here to present our next solution for your self-QC on the next slide. Alex, next slide, thank you. So-called Dipstick solution, which comes with the fourth generation of the light kits. So with this kit, we eliminate all the pain related to the PCR and electrophoresis. So practically you don't need the equipment. You simply need one thermoblock, which you anyway need to perform affinity selection of your Delft self-served kit. It's easy readout, it's user-friendly, it has high accuracy and sensitivity, and in the same manner as the old, let's say, QC, it reports levels of cross-contamination, and it also reports if you have adequate number of Dell molecules in your probe. So if we take a closer look on the scheme on your practically left side, where you see X, practically you can see 3 bands, and practically the third band marked with X is actually 1 band too much, and that means you have a cross-contamination in that sample. The stripe or this dipstick with number 2 has only 2 bands. It has a control band, which is blue, that means everything worked great. And also the pass, the red one, is of the appropriate intensity, everything looks fine. The third one, actually, it looks, you barely see the red band, which means that you highly likely have too low concentration of the Dell molecules in the sample, and that you should not practically send this sample for processing. And the fourth one also looks well. We have everything what we need to see. So this how easy is the readout. And now, of course, is the next question. So how we have to prepare the samples, how we come from these post-selection samples to this dipstick. So next slide, please. So this is really easy process and easy readout. So you get a very, let's say, very nice description how you perform the assay, but basically you get all the reagents in the tubes, you add them to your samples, you incubate your samples for 5 minutes at 37 degrees, and your samples are ready to be loaded on these dipsticks, as you can see on the middle image. And then you slowly see how practically the front travels. And as soon you see the blue line, the reaction is completed, and you can actually practically, you get the results of your QC, and you see if your samples pass the QC or not. And this is actually shown now on the third slide. So we are really looking forward that you will test this type of sample QC, and yeah, looking forward to your comments.
All right, thank you, Alex and Nuska. Next up, Ken is going to join us to talk about his experience using Dell along with a few other testimonials.
All right. Let's see, can you see it now?
Yep.
There you go. All right. Hello, everybody. I'm Ken Westover. I'm from UT Southwestern in Dallas, Texas. It's great to be with all of you today. So today I'll tell you about our experience with the Dell Open platform for the discovery of RAS inhibitors. And as some of you may know, RAS inhibitors against the KRAS G12C mutation were recently approved for the use in lung cancer. And this is really exciting news for the RAS field and a segment of lung cancer patients. But these inhibitors only work against the G12C mutation. And so there's a lot of additional work to do for other RAS forms. And so I'm going to talk about our efforts in that regard. So as background, RAS is one of the most commonly mutated genes found in cancer and is enriched in lethal cancers such as lung, colorectal, and pancreatic cancer. Prior to the acceptance of these G12C inhibitors I mentioned, there was some question as to whether or not RAS was actually a good drug target. Part of this is because RAS or farnesyltransferase inhibitors failed in the 1990s, and this was a large effort by the RAS field to target RAS. But another observation was that RAS mutations are found early in carcinogenesis and were thought to maybe not be required for tumor maintenance. But multiple genetic studies now, and also the results of the KRAS G12C inhibitor trials in humans now strongly support RAS as a therapeutic target. This slide just highlights that KRAS G12C, which is in a black box, in the small black box in the middle there, is just one of many RAS mutations that drive cancers. And this data is replotted from a review by Chris Counter and collaborators showing how specific mutations are differentially and non-randomly enriched in a number of RAS-driven cancers. The mutation frequencies are reflected by the colors, the shaded boxes. Amongst the three major RAS isoforms, there's 3 of them, and they're about 85% identical. The top threshold is 10%, although some really common mutations like G12D, for example, are actually much higher than 10%. But as you can see, we have a lot of ground to cover in the RAS space. There's a lot more we have to do to target other RAS forms, not just G12C. And so we think that the Dell Open is a great way to go to find some new inhibitors. Now, the key to targeting RAS is to understand how it really works at a deep level. So in many cases, we have high-level details about how the components of the RAS signaling pathway, how they're logically connected. And this general understanding has motivated development of inhibitors against many levels of the pathway. So we have EGFR inhibitors that are in clinic. We have BRAF inhibitors that are in clinic. Inhibitors against MEK and ERK, they've all been tested in clinical trials. However, the central node of this pathway, RAS, which is seen in green at the middle, has not been targeted until very recently, despite being one of the first oncogenes discovered over 40 years ago. And this long history reflects the difficulty of understanding how RAS actually works to pass integrated biological signals from the cell membrane to the nucleus. In reality, RAS signaling looks more like this, where RAS signals from within these large multimeric protein complexes. And the architecture of these complexes has been a complete mystery until somewhat recently. And this is because these complexes are quite dynamic, in fact, and can't be isolated easily for structural study. So my lab has been focused on understanding the structure of RAS complexes for the last several years, and we're starting to make progress. And this is a representation of a large RAS assembly on a membrane that was just published last month, or this month actually. We and collaborators used molecular dynamic simulations, experimental structures, and other biological data to assemble this model. So what does this have to do with RAS inhibitors? Well, essentially, our current view is that KRAS G12C inhibitors, which are allosteric inhibitors, essentially break apart this complex. So we think it's possible probably to find new compounds that can also bind to other forms of RAS, not just G12C, and thereby break apart the signaling complex. And we think Del Open is a great way to find tight binders that can do this, shift these dynamics. To understand how to allosterically inhibit RAS, you have to understand the structure of RAS and how it moves. Under normal circumstances, a nucleotide, the guanosine phosphate shown here in blue, strongly influences the structural state of RAS. And specifically, there's a third phosphate, the one in red. It forms interactions with this structural element called switch 2, which is in green. And this pins switch 2 to the body of the RAS molecule. However, if the phosphate's lost through enzymatic hydrolysis, then switch 2 becomes unconstrained. And you can see on the right-hand side how it swings it open in this structure that was solved. And this creates an open pocket adjacent to the guanosine diphosphate. And this is, in fact, the pocket where KRAS G12C inhibitors fit. So interestingly, this pocket is quite malleable, as you can see by comparing two of the clinical RAS inhibitors that are now well studied. So on the left, you can see adagrasib kind of shifts switch 2, so it's rotated laterally, whereas sotorasib, switch 2 is kind of shifted upwards. And this just emphasizes the malleability of this induced pocket. So we want to find additional allosteric inhibitors that either bind to switch 2 pocket or other parts of RAS. So RAS actually moves a lot, providing many opportunities to target other pockets. This is a morph between multiple structures that we and other labs have solved of RAS, showing how switch 2 and switch 1, which form part of the nucleotide binding pocket, how these move quite a bit. And I think what you can appreciate here is that there are not only opportunities under switch 2, but also opportunities underneath switch 1 to bind small molecules. So we, in seeking, molecules that could bind in these induced pockets, we did a series of Del Open screens to find new direct binders. And we did this in a way that wouldn't bias toward any particular pocket. And this resulted in a list of hits. We got 5 that we had made, actually 10 for two screens that we did in total. And then we went on to test these to try and validate them. So our first step in validating these hits, since we didn't know where they bound, was to do differential scanning fluorimetry to look at the impact of these compounds on the thermal stability of the proteins. And this technique's really fast and simple and easy to adapt to the various RAS forms we were studying. Essentially, the principle is that you use a fluorescent dye represented by the green molecules that emits a signal when it interacts with hydrophobic residues that become exposed as a protein unfolds when you heat it up. And then small molecules, which are depicted in the black circles, these increase the stability of proteins, so they're more resistant to heat denaturation. And in this way, you can begin to validate some of your hits in a quite easy way without knowing where they bind. So of the hits we received from Wuxi, several showed an impressive ability to increase the thermal stability. And so on the left, you can see raw fluorescence traces. And then on the right, we're showing the first derivative, which kind of gives you a better sense for where the temperature peak falls. For this type of experiment, a change of 4 to 5 degrees is generally significant, gives you some confidence. But in this case, we're getting almost 20 degrees of change, which is really impressive. So although I would love to go on and on about this, I can't do so at this point, but I'll say that at this point, we've been able to solve co-crystal structures of these validated compounds. And here's one of the very recent data sets, actually, that shows beautiful diffraction out to about 1 angstroms. And we're working through these structures now. But I would say this has been a very successful effort for us. So in summary, we've been really pleased with our collaborations with Wuxi in running the Dell open screens, and we've found promising leads against new forms of RAS. And we're looking to further develop these and do additional collaborative work. And then last, I just wanted to acknowledge the people in my lab, especially Lamba Lee, who was instrumental in doing the Dell open screens.
Hello, this is Ken Westover from UT Southwestern Medical Center in Dallas, Texas. Our lab has been using the Dell Open platform to discover inhibitors of major cancer drivers such as RAS and kinases.
Hi, this is Xiang Liu from School of Pharmaceutical Sciences, Tsinghua University. Our lab has been using Dell Open platform to screen for novel drugs targeting G-protein coupled receptors, which are major drug targets for many diseases, including heart disease, cancer, or diabetes.
大家好，我是上海中医药大学徐建荣老师。我们的主要研究方向是胆碱能系统在神经退行性疾病中的调控机制及相关药物研究。对 GPCR 受体而言，因为调控方式的多样性与下游信号转导通路的复杂性，使得寻找具有合适药理活性的先导化合物极为困难。戴尔筛选作为一种重要的先导物寻求手段，它具有比较高的进入门槛，对于小型课题组来讲比较难以直接应用。但得益于戴尔 Open 平台提供的非常开放性的合作方式，我们多了一种新的可能，我们可以直接应用戴尔方法对我们的靶点进行筛选，使我们能够搜寻的化学空间得以大大的扩大。
Hi, my name is Joe Matarlo. I'm a senior scientist here at Pledge Therapeutics.
Hi, I'm Asanga Bandara. I'm a computational chemist at Pledge Therapeutics.
And we use Dell to interrogate targets that are difficult to drug in the fields of immuno-oncology and virology. I perform the screens.
And I perform the subsequent data analysis and cheminformatics.
大家好，我是复旦大学药学院博士研究生。我们现在正在用药明康德的 DER Open 这个技术进行药物筛选工作，期待会有一个好的结果。
All right. Before we move into the Q&A portion of this webinar, I'm going to pass it over to Alex for a few more words.
Okay, thank you. Let me share my screen then. Okay. Oop, get rid of that. All right, so, oh, that's not the right. Here we are. Okay, so the last bit is about something that we've generated recently. It won't be available for a few more months, but we wanted to go ahead and just sort of float it out there so that those of you with an interest in this can sort of prepare yourselves for this coming available in the near future. So what we have is a home ASMS kit. And so what this would allow you to do is to get your hits and to test them for binding to your target, but you wouldn't need a mass spec or to develop the expertise for this. So we would send you the kit. You can do some simple steps in your own lab to determine binding against your target. Again, you don't need to share what the target is with us, so you can keep that a secret. And then you can send the lab samples back to us, and we can tell you if indeed you have found true binders to your target. So that's just one slide I wanted to present there very quickly before the Q&A. And now, Arsalan, we can we can go back to the Q&A.
All right. Well, wonderful presentation from y'all. Thank you so much. And we have so many questions. Let's try our best to get to them all. Let's start right off at the top over here. Question for our panel here. Can you elaborate more on what data is shared in the Dell Open model?
Yeah, so for Dell Open, when you first do the screen and we sequence the data and we get the results, we share really all of the data with you except for the structures themselves. Part of that data includes the physical property profiles for each of the hits and the enrichments. And of course, in the future, we'll include the frequent hitter index as well. And using this data, you need to make the choice about which 5 hits you would like to follow up and have synthesized, even though you won't know what those exact structures are for those hits. Now, of course, at any point in time, you can find out what the structures are. But in order to do that, you would have to tell us the name of the target that you screened. And then you need to give us permission to post all of that metadata for the screen minus the structures on the Dell Open website. Dell Open it's overseen by an academic group, and all the different academic groups, they all have to obey these rules that they have set up. Yeah, so that's how the data sharing works with Dell Open.
Okay. All right. Are the new libraries in the 4th generation kits only in the Dell Lite kits, or are they also in the Dell Open kits?
So there are new libraries in both kits. So there are libraries now in Dell Open in the 4th generation that weren't in there before. And now with that said, there's more new Dells that have gone into the 4th generation Dell Lite kit than in the Dell Open kit.
Okay, very good. Here, I'm going to read this question. There's several questions packed into here over here, but we can read it over here. Thank you, Juliana. Here's the question. Thank you very much for a great presentation. If I am not mistaken, in each kit, you have 58 Dells. What kind of information do you provide to the users about each library in case users do not want to screen all of them and select only a few of them that will fit their needs?
Okay.
You also work on focus Dells. So why don't you answer that first?
Okay, let's start
question.
with that.
Okay.
Start with that. So very often we're asked, we have these Dell pools, whether it has 58 Dells or 20 or 100, and people ask, can we just screen certain Dells? And over again, both here at Wuxi and at other places, what we who practice this have found is that it makes no difference for the results if you pull out certain Dells and you exclude others. The screen is done all at once. It's done in a single tube. And so the short answer to your question is, for a Delight kit, you screen them all and you screen them all at once, and you can't cherry pick which ones you would like to screen. If we thought there was a benefit to it, though, we would set it up where you could, but we can't find that to be true. Okay.
That probably answers the rest of the question too, because the rest of the question is, do you work on focus Dells? How does the process of focused library design look like? Do you provide a customized Dell design service?
Well, that's a little bit different. So let's say you have a fragment and it binds to your target, but weakly. You've tried to do some med chem and you've gotten nowhere. Why not come to us, and in 6 weeks, we can make hundreds of thousands of versions of this fragment and screen them in our binding assay for the Dell screen. So the short answer is yes, we can do that. If you have a fragment that is unique and it's a weak binder and the medchem SAR just isn't working well, we can build a novel fragment-based Dell around that fragment and screen it for you.
Okay, all right. Let's get to this question. For Dell Open, what would be the approximate time frame from application for the kit all the way to receiving the selected 5 compounds for validation?
So when we get the samples back, it takes us, I believe it's 4 weeks to return the results to you, at least for Dell Lite. For Dell Open, the time span might be a little bit longer. It's not guaranteed quite as much, but it's, Nuska, would you say it's safe to say like 1 to 2 months for a Dell Open?
Well, yeah, usually we process all the samples about at the same speed. So it's about 4 to 6 weeks also for Dell Open. Yeah.
Okay. All right, I think this question is for Ken. So what's the benefit for the user to use the 5-minute QC instead of using a traditional qPCR? Is there a special consideration of such design? Is that the question you want to take? Who wants to take that question?
I think that's more for Nuska.
Okay, yeah. So, yeah, thank you for this interesting question. So the qPCR step is actually one of the crucial let's say, sample inspection quality control. That means it has to be performed under great care, under standard conditions. And the people who perform this step really have to perceive extensive training so that this is highly reproducible. Only this actually guarantees then the proper sampling of the this DNA libraries which you have in your post-selection samples, and practically enables the best possible quality of your screening data. And we don't want to put this heavy load on the shoulders of the client, which are using our kits. So that's why we decided that for them, we provide a very simple, very easy method of quantification of the samples, which practically give them a yes or no answer. That means, yes, I have no contamination. Yes, I have enough, let's say, copy numbers in my sample. And all this complicated part is then done by us. And when we sample, then of course, the probes accordingly and send them for next, so to say, to the sequencing. Yeah, it's simply to be more user-friendly and so we do the tough work then.
Okay, very good. Another question over here. Does a client have to partner with Schordinger on the Dellite machine learning approach, or could they apply their own algorithms?
Yeah, so when you unlock all of the structures for Dellite, you unlock all of the structures that have enriched above a very low threshold. So you will get hundreds of thousands of different structures, maybe even more, when you unblind that data. And you could perform, yeah, any kind of analysis on that data that you would like within your own labs, and you don't need to tell us about that. And the problem is you won't have the negative data. So if you need to know all of the structures of the hits that didn't enrich, well, there's no way for you to get those through the data set that we unblind. So if you believe that that data set is needed in order to do the deep learning, then yes, Schordinger would be who you would have to partner with.
Okay. Here, let's go to another question over here. Here's the question. I have already screened using a Dell Open kit and have gotten hits. Do you recommend I rescreen using the new kit since there were new libraries added?
I guess I probably would. Yeah. If you have enough protein and you have willing graduate students, and all graduate students are willing, right? Then yeah, it seems like no harm done to at least try it out. Yeah.
Okay. Another quick question for you guys. We have just under 5 minutes left. I'm gonna try to get as many of these in as possible. Quick question. Can my target sample contain DNA? Example, DNA binding protein plus DNA.
Yes. Yeah. So if you're screening a target that's a DNA binding protein, probably the best thing you can do is block that binding pocket with DNA of your choice that you know is going to block it effectively. And yeah, that's perfectly fine. No problem there whatsoever.
Okay. A question on rights. How about this? Which rights does Wuxi AppTech retain related to Dell Open, related to the develop of competing compounds to the information obtained through the Dell process after sharing target info?
Ooh, you might have to read some of that again. So it was about the rights that Wuxi has, or the rights that
which rights does Wuxi retain related to Dell Open, essentially?
None. So you get the 5 hits, we share the structures with you, and then you're just free to do whatever you want. And if you make $1 billion, we don't come by later and say that we have some right to it. Although it's a good idea to try, maybe, but that's not how it is supposed to function.
Very good. Very good. A couple more of these questions. Here's a quick question. How do we get more info of more than 5 hits in Dell Open?
Well, at the moment you sort of can't. But with that said, let's say we share the structures with you and your data is put on the Dell Open website without the structures. If another collaborator comes along and wants to work on some more hits from your data set, they could unblind another 5 hits. So there is a way to get more structures from that data set, but it requires collaborations with other professors.
Very good. Let's try to get a couple more questions in here real quick. I've noticed that all of the libraries in the Dell Open kits contain peptide bonds. What's the advantage to having peptide-like libraries, since peptides are not generally drug-like?
Well, there's all sorts of drugs that have an amide bond in them. So I believe we showed you one example where the hits wouldn't necessarily have any amide bonds. So there are certainly structures within the Delight that have no amide bonds. But with that said, yeah, you can have an amide bond and easily fit within even CNS space. So I wouldn't really agree with that statement.
Okay, all right. That's a good pushback over there. Another follow-up question. How do you decide which structures to add into the kit? The kit is made up of focused libraries of compounds or diverse libraries of compounds?
Completely diverse. It's completely target agnostic. So our goal is to, like, if you look at that 3-dimensional plot of chemical space, the goal is to fill up as much diverse space as possible. So that's the primary goal. It's not to focus in on any specific fragment or existing drug.
All right. Okay. That's about all the time that we're going to have for today. I want to point everyone in the audience, if you want to contact Wuxi AppTech, in the chat, we've put an email address for you over there. Thank you, Alex Sachs. Thank you, Nishka Shammer and Ken Westover, for your expertise today. Thank you, everyone in the Endpoints audience, for joining us. And we appreciate Wuxi AppTech for sponsoring this discussion with Endpoints News. If you would like to rewatch this webinar or share it with colleagues, a link for on-demand viewing will be made available tomorrow. I'm Arsalan Ar for Endpoints News. Thank you for joining us, and we hope to see you at a future webinar. Thanks, everybody.
Bye.